Hello, welcome to our mitosis lecture with PowerPoint. So we did this today in class and it went pretty quickly. So like I said, it is a biology review, so we're going to go through these fast, okay? These are the notes that you're going to put on this page. Let's see if I can get it in there. It says the cell cycle mitosis, okay? So if you remember, mitosis is splitting of one cell into two identical cells, okay? Mitosis, I remember this by the T, I get two totally identical cells. So meiosis is where you get some information from a daddy cell, information from a mama cell, and they come together, and then you get cells from there. But that is where those genes mix, right? And they kind of go through mitosis twice in that mix. So mitosis, we're not even doing meiosis now. We're doing that later when we talk about like sexual reproduction and stuff. So mitosis is just one cell splitting into two identical cells, okay? As you fill in the notes, you just fill in each box. As we go, you might have to write small. You don't have to write every single little thing. Go ahead and condense it to how you will understand it when you go back to read your notes. Okay, so there was a YouTube video that we watched. It was really quiet in class. I'll add that. It's the lower one. I'll add that to the web page. If you want to go in and watch that, I'll include that. Okay, so interphase is a phase and it's not really a true phase of mitosis. Mitosis is actually from prophase through telophase, but this interphase is an inner period where the DNA is being copied and the chromosomes in the nucleus have two connected copies of long, stringy sister chromatids. And I kind of look at it, think of it like spaghetti, just kind of all in there squiggled around together, okay? But it's where DNA is copied, and then also the centrosomes are copied. So these kind of things here with the lines coming out of it, those are centrosomes, okay? And those have been copied. So normally in a cell that's not ready to reproduce, not ready to split, you know, to make more, you just have one of these, okay? Now we've got two. We've copied the centrosomes and copied the DNA. It's kind of like an organization period. They're just getting started, getting things organized. The next phase, this is where the cell enters mitosis, and it's called prophase. I remember this, the P in prophase reminds me that it pairs up. So here the chromosomes condense. So we don't have those stringy spaghetti type things. We now have X's. We can actually see some sort of a form of coming really condensed and getting X's. The nucleolus fades, okay? The spindle grows between the centrosomes. So those centrosomes on the sides, they start shooting off these spindle fibers, and they grow and kind of go to attach these chromosomes. Okay, and the centrosomes go to either side of the cell. Prophase, pairing up. Metaphase is where those chromosomes meet. They all meet in the middle. So all of them will come to the middle of the cell, meet up. You still have your X's, okay? And at this point, something really interesting happens. It's called a spindle checkpoint. So it just does a little quick check. Are things working correctly? If we do go to pull to split, does it seem like everything's going to split correctly? If not, then mitosis stops until the cell fixes the problem. So here is a point where it does a quick check. Is everything in check, ready to still go forward? If it is, then we will. So metaphase meets in the middle. Then you have anaphase. This is where they come apart. So you can see where they used to be X's and now they're being pulled apart in more like V's, okay? So anaphase apart. These sister chromatids are then pulled apart into separate chromosomes. They've been separated and they pull to those opposite ends. So those spindle fibers are like pulling them back. This is also a time where the cell will elongate a little. Okay, so instead of like this, we're getting like this because things are pulling apart. We need a little bit more room, right, to get those pulled. So anaphase A apart. And then the last part is telophase, and I put this with it tears. It's tearing. So you might even think of it as pinching. So the two sides will start to pinch and come together, and then eventually the tears, okay? The spindle is broken down. So remember all those stringy spindle fibers that we're pulling, those break down. You get to 2 new nuclei will form around those chromosomes now. The nuclear membrane and nucleoli reappear, and then the chromosomes decondense. So remember, we started out with like spaghetti, like stringy. They condense and get to be X's. They pull apart as V's, and then now they start to decondense and become stringy again. Once this complete middle is torn, okay, once it completely pinches away, then we have the division of 2 cells. And so cytokinesis is not a phase of mitosis, it's a process. So cytokinesis actually means cytocell kinesis formation. So we're forming a cell here, and at this point 2 cells, and it's the division of the cytoplasm that forms 2 new cells. It may start in anaphase at that elongation stage, or telophase, where whenever it starts to pinch or furrow, that's really where cytokinesis starts. And once you've got 2 different cells, cytokinesis has happened. You have 2 new cells. And that's at the end of telophase, when that final tear happens, cytokinesis. 2 identical daughter cells. On the side of your page, so that's upright, on the side over here, there's a place where it says 1 cell divides into 2 genetically identical daughter cells, okay? So that is your quick review of mitosis. I had the students in class just turn their page over or just get a blank piece of page, and then I had them write down the little acronym or word to remember the phases in order, and we didn't do that here. So if you put it all in order, it's IPMAT, I-P-M-A-T, IPMAT, okay? And then it stands for interphase, prophase, metaphase, anaphase, telophase. So since I just told you them in order, I want you to maybe give it some time and then go back, maybe for a bell ringer for the next one. Go back and write down, see if you can write down that word to remind you of the order, and then write down the actual phase. Okay, what does the I, what is the P, M, A, T, what does those stand for, the actual phases, and then what is the reminder word that starts with those also. So for example, we don't have one for I, that's just kind of the organization phase, but P, prophase was pairing up, right? The P is for pairing up. So go back and write those next to it so you can just remember the order, what it, the phase, and what the phase means. For the test, you'll need to, the worksheet has it too. It has pictures of those phases, and you have to match up which one goes to which picture too. Okay, that was it. Very sweet and simple. How long was that? 9 minutes. Okay, done. At least 5 more points in your notebook for notes. See ya.